All right, so all of that we had just talked about is all of the description of the structures of the conducting pathway. All of those structures that bring the air to the lungs, treat the air, filter it, moisturize it, or humidify it, I guess I should say, raise the temperature up so we could get it to those gas exchange surfaces deep in the tissues of the lower respiratory system, deep in the lungs. So from the terminal bronchioles, we branch into respiratory bronchioles, and then from there, each one of those will actually branch into several alveoli, which are these sort of bag-like structures right here. The alveolar duct, which you can kind of see if you zoom in, let me get a different color here for y'all, right here, ends at what is known as the alveolar sac. So here's that space right there. And the alveolar sac has it comprises a series of alveoli. Now the singular is alveolus, so that's what these little bubble-shaped looking things right here. And each one of those sacs is surrounded by elastic fibers to help hold the sac open. Here's another diagram. This is a scanning electron microscope. That's what SEM stands for, of the those exchange surfaces or structures in your lungs. And so you can see the spaces right here are the actual alveolar sacs. You can see let me back up because I think I just made the label. So here's our alveolar sacs. Here's our big space right here. We've got alveolar ducts that terminate at the alveoli, which are those little bubble spaces right there. All of these little pockets, essentially. So if we take a little bit of a closer look at the alveolar surfaces themselves, you can see some important structural characteristics. The lining of the alveoli is one cell thick, and it's primarily these cells called pneumocytes type 1s. Now there's two pneumocytes. Pneumo is referring to lungs, and cyte is cell, so you could think of these as lung cells. So the pneumocyte type 1 is where the actual gas exchange takes place between the blood in the capillaries, which you can see these big red blobs right here, and the alveolar space. So the blood, the capillaries within the lungs bring carbon dioxide rich blood to those spaces. That carbon dioxide diffuses out of the capillaries, goes into the pneumocytes. From there, it is exchanged with the oxygen that's in the alveolar space there, the alveolar sac. Then the oxygen comes back in, going the opposite direction. And then you also have these other cells right here. These are the pneumocyte type 2. And these cells are responsible for making a substance known as surfactant. Now, surfactant, you probably are a little bit more familiar with surfactants than you think about. Surfactants are an oily secretion secreted by the pneumocyte type 2 cells that primarily are phospholipids and proteins, and they coat the alveolar surface to reduce surface tension. So this is a very liquid environment where we have an interface between the liquid environment of the body and the air environment of the atmosphere around us. And so anytime you put air and liquids like water, something like that together, you form bubbles. Now the surface tension of water in the liquid environment that you're creating is actually strong enough that you can collapse these air spaces just with the formation of these bubbles. And so the surfactant that is produced by the pneumocyte type 2 cells reduces that surface tension and allows those elastic fibers to hold those alveolar spaces open. If for whatever reason your surfactant is compromised either by injury or genetic abnormality or something like that, you have something known as respiratory distress syndrome, which basically means those alveoli collapse every time you exhale because you have those bubbles that are forming, which can be treated based on the source. Treatments are going to be different. So that surfactant is actually kind of important. Now, one thing that's interesting that I've learned in years past is there's a lot of research that's sort of still going on that looks at the importance of surfactants. And so there's a lot of really cool research going on with how those works and modifying those, and how if there's particular chemicals that you inhale that can actually interfere with the surfactant's ability to perform its function. The actual process of gas exchange takes place at the blood-air barrier, which is this section right here. Okay, so we have this space right up here is the capillary. Here's our giant monster red blood cell. We have the capillary endothelium, so that's that inner layer of the capillary. And remember, your capillaries are also one cell thick to allow these substances to fuse across the membrane of that particular cell. And here's the nucleus of that cell. This is the lung side. Right here, we have the alveolar space, that empty air pocket. Here is the cell layer of the alveoli, and then we have a basement membrane in between the two that holds them together. And so the three layers, when we're talking about the blood-air barrier, let me clean up some of this stuff real quick, is the alveolar cell layer, which is the purple right here, the fused basement membrane in between the alveolar cell layer, and the capillary endothelial layer. And so this allows air molecules, carbon dioxide and oxygen, nitrogen also comes across as well, to come from the outside air into the bloodstream and vice versa, but it blocks other larger particles like dust from coming in or proteins from coming out and stuff like that. This process is really, really fast because the distance for diffusion, we're talking about two cell layers basically, and in that those cells are very, very thin. So the diffusion distance is very, very short. O2 and CO2 are both very small molecules. They're lipid soluble, so they pass through the cell membranes very, very quickly. Now, if we have inflammation of the tissue, specifically at that blood-air barrier, that is known as pneumonia. This inflammation can lead to fluid leaking into the alveoli, because part of the inflammation process is the buildup of fluids. That's where the actual swelling takes place. And so as the fluid floods into those really small tissue spaces, it can actually be forced into the alveolar sacs. And this compromises the blood-air barrier's function, so oxygen is not allowed to come in, carbon dioxide is not allowed to come out. If you've ever had pneumonia or known somebody who's had pneumonia, it's characterized by having trouble breathing. So kind of similar to that bronchitis feeling, it kind of feels like somebody's sitting on your chest because those passages are filled with fluid. Now remember, bronchitis is different. That's just inflammation of those conducting pathways, restricting airflow. This one is filling of the air passages, preventing gas exchange from taking place. And also it's a bad deal because a bacteria can live in that fluid. It's very difficult for your immune system to purge any pathogens in there. So pneumonia it can be very serious, especially if you have underlying health conditions, you're elderly and stuff like that. All right, let's do a little bit more conversating about anatomy. We've got a few more slides. We'll talk about the actual gross anatomy of the lungs. All right, so when we're talking about the lungs, humans, mammals in general, humans in particular have left and right lungs that are sort of connected by way of the bronchioles, the bronchi, sorry. Those are both in left and right pleural cavities, and you can see this tissue right here, which is actually a boundary that separates the pleural cavities into two separate left and right pleural cavities. And then the inferior portion, also known as the base of the lungs, right here, sits on a thin shelf of muscle that's called the diaphragm. So that's what sits like here-ish or so. The lungs are separated into separate lobes, and we kind of looked at that when we looked at the airway branches that go into the different lobes and stuff like that. And so the right lung has 3 lobes, the superior lobe, the middle lobe, and the inferior lobe. And those lobes are separated by fissures. One is the horizontal fissure, and then the other one is the oblique fissure. And the left lung has 2 lobes with 1 oblique fissure, so it only has the superior lobe and the inferior lobe. The right lung, so looking at this side right over here, is wider than the left lung, and the base right here is actually displaced by the liver. The left lung is longer than the right lung, and the medial margin right here is actually indented, and that space is known as the cardiac arch, and that indentation is formed to make room for the left ventricle of the heart. Remember, the heart is going to sit sort of here-ish. And there's the vessels. And I'm sorry, it looks more like a pineapple than it does a heart. But your heart sits right here. The left ventricle is going to fit into that notch like a little pocket. And then you've got your liver, which is sort of vaguely triangular shape. That sort of sits, I guess it's more like this. That sort of sits like that underneath the lungs. We have a region in the lungs kind of towards the center that is known as the hilum. It's a groove, so we've got a cutaway section right here and right here that allows passage of the bronchi as well as the pulmonary veins and the pulmonary arteries to actually enter into the interior portions of the lungs. And then we have the, within the hilum is the root of the lungs, which is a series of dense connective tissues along with nerves and other vessels that anchor the lungs to the mediastinum. So if we go backwards here for just a second, your hilum, you can't see it from this view. It's actually going to sit sort of in this area, and the vessels coming out of the heart are going to pass into there, and then they're going to pass out to go back into those atria to complete the pulmonary circuit. Within the lungs, we have tissue known as trabeculae. Trabeculae are the fibrous partitions within the lungs. They contain elastic fibers, smooth muscles, and lymphatic vessels as part of the immune system, sort of drainage system, basically. These divide the lobes into smaller and smaller compartments, and the pulmonary lobules are divided by the finest partitions known as the interlobular septa. So the pulmonary lobules are the smallest divisions of the lungs, bound by these fibrous partitions. As far as the blood supply is concerned, this ties into some of the stuff that we talked about with the heart. So where we have those gas exchange surfaces, those surfaces are going to receive deoxygenated blood from those pulmonary arteries. At that point, gas exchange is going to take place across those capillary beds that surround each alveolus. From there, once we have the exchange taking place, so the blood becomes oxygen rich, it leaves through the venous end of those alveolar capillaries, eventually collecting back together into the pulmonary veins, bringing the blood back to the left atrium. From there, left ventricle, heart, of the heart, aorta, systemic circuit, and then back. Bronchial arteries within the heart bring oxygen and nutrients to the conducting pathways by way of the capillaries. In the pulmonary circuit, so we want to talk about blood pressure in the pulmonary circuit. I apologize. The blood pressure in the pulmonary circuit is lower than the systemic circuit. And we kind of looked at that a little bit. That right ventricle is a lot smaller. The amount of force required to move the blood is a lot lower. So that ends up being lower blood pressure in the pulmonary circuit than in the systemic circuit. A lot of heart diagnoses and stuff like that, when they're looking at cardiac output, they're primarily looking at what the left ventricle is. However, because this blood pressure is lower, it's easier to block the pulmonary vessels by fat deposits causing clogging of the arteries, blood clots, air bubbles, and blockages of these pulmonary arteries are known as pulmonary embolisms, where we've blocked one of the branches of the pulmonary arteries that stops blood flow to the lobules and the alveoli, and then those tissues can start to die, and that restricts your gas exchange capacity. There are, like I said, there are two pleural cavities. So here's a cross-sectional view of the thoracic cavity showing those pleura. The pleura is the name of the tissue that surrounds the lungs. An analogous structure would be the pericardium that surrounds the heart. The two cavities are separated by the mediastinum, which is the cavity that holds the heart. So the two pleural cavities are not continuous cavities. Each cavity is lined with a serous membrane, which is known as the pleura. The pleura has two layers: the parietal pleura, which is illustrated right here, and the visceral pleura, which is an inner layer right here, and also serves as the outermost tissue boundary of the lungs proper. In between those two layers, the parietal pleura, which also the parietal pleura lines the inner thoracic wall. It has a pleural fluid. Let me write that for you. Which is in between the visceral and the parietal pleura. And that provides lubrication for that space in between those two tissues to allow movement for the lungs during breathing and twisting and stuff like that. I think we'll go ahead and say that this video is over. The next ones that we're going to be looking at are the actual physiology of how it works. So in the next video clip that you're going to watch, it's going to talk about external and internal respiration with basically the mechanics of breathing, defining what external respiration is, how that's different from internal respiration. And then as we get through that one, that'll segue into how gas exchange takes place. We kind of talked about that a little bit when we did cardiac physiology. So we'll get into that in a little bit greater detail with that third video. And then that third video will round out this sort of mini-series with changes that your lungs go through structurally and physiologically over time as you get older, the effect of inhalants like smoking and stuff like that have on your lungs, and some of the interactions with some of the other organ systems that we've looked at. If you need to go back and go through this to take notes, you can refer to the slideshow the slide notes that I created if you need to. If you have any questions as you're going through this, let me know. Send me an email, send me a remind message. Otherwise, I look forward to seeing y'all again soon.